I welcome you all to my lecture. I'm Ms.T.R.Sambavi, Assistant Professor, Department of Industrial Biotechnology. I'm here to teach Enzyme Engineering and Technology subject. Our today's topic will be solid state fermentation. So in this lecture, today we are going to talk about the introduction of solid state fermentation and major factors that are influencing the solid state fermentation process. That is the selection of microorganisms that we are going to use for your solid state fermentation and the substrate characteristics and pre-treatment. So how we are going to, on what basis of the on what characteral basis we are going to choose the substrate and what are the pre-treatment strategies that we are going to use for the substrate. And then we're going to talk about the biomass measurement and the environmental factors that is related to the solid state fermentation process. Okay, then we are going to talk about the advantages of solid state fermentation, disadvantages at the same time, and also the application of solid state fermentation. So these are all the topics that we are going to cover for today's lecture. So let's get started now. Introduction about the solid state fermentation process. So what is the solid state fermentation? So SSF consists of microbial growth and product formation on solid particles with minimum amount of water containing sufficient moisture to allow microbial growth. So the solid state, in the solid state fermentation process, we can categorize into two parts. One is that there will be one microbial growth, and at the result of this microbial growth is going to give a product formation. Okay, this is going to happen on a solid particle. So in general, I've spoke about this fermentation process. So what are the types of fermentation? We were discussing about it in the previous class. So we saw about submerged fermentation, okay, which is happening in the liquid medium. Now we are talking about solid state fermentation. So it is going to, as the name gives, solid state fermentation. So it is going to happen on, the product formation is going to happen on a solid particle. Okay, so this is going to be the basic, this is a basic explanation for the solid state fermentation process. Okay. So now SSF is a biomolecule manufacturing process, and the product is used in food, pharmaceutical, cosmetic, fuel, and textile industries. These molecules are metabolites produced by microbes grown on the solid media. So the solid state fermentation is, it is simply, we said there is going to be a product formation in solid state fermentation, isn't it? So this solid state fermentation is also called as a biomolecule manufacturing process. Okay, so the product formation is actually going to be a biomolecule, and this product is used in various industries. So various industries that is, related to biotechnology. Example, it is used in food industries for food processing and other processing and all for food processing, and then in pharmaceutical industries, in cosmetic, in fuel. So in general introduction class about enzymes, we saw a brief process about this biofuel industries. So, and it is also used in textile industries. So this is the various application that we are going to see at the end of this section. So basically this solid state fermentation process is manufacturing any biomolecule. So this SSF employs natural raw materials as the carbon source for fermentation. So what is this SSF? There should be some start for any process, isn't it? So the natural raw materials, that is, we call it as a substrate. Okay, so we have spoke about all the basic terminologies that is related to enzyme technology in introduction class itself. So I have said what is a substrate. Fine. So now this raw materials like any biostream waste, this substrate for your any product here, that is the substrate that you are going to use for your solid state fermentation. It can be of several types. So it can either be a biological, it can either be from biological stream, or it can be from non-biological stream also. But many sources they use is bio waste stream. We cannot exactly call it as biological waste, but we can call it as bio stream waste. So such as this barley, wheat bran, rice bran, sugarcane, and all these oil cakes and all. So after food processing, there will be some waste. So we can also use these waste as a substrate for our enzyme or any product formation here. So I've just given the pictures of various biomasses or various substrate pictures that biological biostream waste. We cannot, as I said, we cannot call it as biological waste. We can call only as biostream waste. So all these waste can be utilized as a substrate for your solid state fermentation process, right? Next, you can also see some examples of the solid state fermentation process. Okay, so you can plan your experiments either in a large scale or in a pilot scale or in a large scale. It depends upon the application, or it depends upon the process that we have designed here. So if you are going to do it in a lab scale, you can do this solid state fermentation in a conical flask or a closed container like this. Okay, so I have just given the pictures of the process here. If you are going to do it in a lab scale, okay, so you can either do it in this conical flask, or you can see it in the top left corner, a closed jar. You can either do it in a conical flask or this jar. And if at all you're planning to do it in a pilot scale, okay, there are a lot of reactors that have been specially designed for carrying out your solid state fermentation. So say for example, like you just wanted to produce cellulase enzyme. Okay, so you are going to employ the solid state fermentation process. You can specially design a tray reactor according to your needs. Okay, so we are going to see those parameters that need to be considered while carrying out this SSF process. So once I talk about all those parameters, maybe you can decide based on those parameters, you can decide what kind of reactor that you can employ for your according to your needs. Okay, so if it's going to be a pilot scale, you can do it in a large trace. And if at all you're going to do it in a large scale, you can see it in the bottom pictures. No, in large scale, in industrial level and all, there will be a sequence of tray reactors to because you need a voluminous amount to be occupied in that. So according to that and application and other process design, you can decide in whatever mode that you're going to do. Okay, so these are a few examples for the solid state fermentation process, right? So now we'll get into these major factors that are influencing the solid state fermentation process. In that, now we are going to talk about the selection of microorganisms. So what is the selection of microorganisms? So in introduction part itself, I said the solid state fermentation is of two phases. One is that you are going to grow some microorganism, and then you are going to focus on a product formation. Fine. So for that, you need to select some microorganisms. There are a lot of microorganisms is there are wide ranges of organisms are there. So according to your need, according to the application that you are planning to perform, based on that there are certain selection criteria of microorganisms. So first, the types of microbes. So what are the types of microbes? Let me know. So there is bacteria, there is fungi, there is yeast, So basically for solid state fermentation, bacteria, yeast and filamentous fungi, okay, can grow on solid substrate and are mostly used in solid state fermentation process. Okay. So as I said, this bacteria, yeast and filamentous fungi, they are mostly these organisms are employed for solid state fermentation process. Bacteria are mainly involved in composting. Okay, so this bacteria, which we said like we are going to use bacteria for the solid state fermentation process. No? So what kind of bacteria? Again, this question will come. So bacteria, which is involved in this food processing and all can be used in solid state fermentation. Why especially bacteria that are involved in these processes are involved for solid state fermentation? Is that because, as I said, maximum will be using any biostream waste for solid state fermentation, isn't it? So since this is based on biostream waste, the nutrients that is required for a bacteria to grow will be there in that substrate. So there is no need for us to provide any additional components for that organisms to grow. So keeping that in mind, the bacteria which are involved in this composting and food processing and all can be employed for solid state fermentation. Okay, next we said yeast. So yeast can be used for production of ethanol, food and animal feed, also have been used in numerous solid. Okay, so about this yeast, we spoke about bioethanol. I think you guys will remember that. That was supposed to be the major topic in the enzyme application. So we spoke about application of cellulase in biofuel industries. Right? So yeast is used for converting your sugar into ethanol. Fine. So for that we are using yeast. So next is this filamentous fungi are the best and used mostly in SSF owing to their physiological, enzymological, and biochemical properties. So this we most probably, if you look into this solid state fermentation process and all, the yeast is the major. What is it? Yeast is going to be the major microorganism that is used. But other than yeast, filamentous fungi are said to be the most widely used microorganism. This is because they have very good physiological, enzymological, as well as biological properties when compared to yeast. So if it is going to be production of bioethanol, then yeast is playing a major role. But apart from that, if you look into the many applications, the filamentous fungi are going to be the major microorganisms that is being used. So what are all the advantages of using this filamentous fungi for bioconversion of solid substrates? So if you look into this aspect, hyphae mode of growth, that is quick spread and coverage. So this hyphae mode of growth, you all know the stages of growth stages of a fungi. So this hyphal mode is protruding part of this fungi, so it will be easy for the fungi to spread wide over. So that's what is mentioned here, the quick spread and coverage. So it is going to spread easily and it's going to cover the maximum biomass that is been occupied in the process. Okay, so high tolerance to low water activity. So this high tolerance to low water activity is that we are already performing a solid state fermentation, and if in case the moisture content is and all very going to be very high, it can easily get contaminated. So in order to the minimal thing that you can do here is to just you can maintain the moisture content and water content in it, so that it will be you can easily prevent your contamination with that. And high osmotic pressure conditions. So these are all the advantages of using a filamental fungi for your solid state fermentation process. And I have also mentioned few pictures here. You can see few pictures here. I've mentioned the Aspergillus niger, and you can just see few penicillium around, few pictures here. So just have a look at it. So now the mode of fungal growth. So now the hyphal mode of growth gives the filamentous fungi the power to penetrate into the solid substrate. Therefore, nutrient can be utilized by the fungi efficiently. Yes. So as I said, we all are like aware of the different stages, growth stages of fungi. So this hyphal mode of growth, well, it is kind of a filament there. So it'll be easy for the fungi to, penetrate into the solid state in the solid substrates. Okay, so it can easily penetrate to the solid surface, and therefore the nutrients can be utilized by the fungi efficiently. So once it can easily penetrate into the surface of your substrate, it will be easy for the organism to utilize all the nutrients from the substrate. So once it is utilizing all the nutrients properly, so what will happen? The fungi will obviously have a very good growth, isn't it? So that is one major advantage here. And then the cell wall structure at the tip of the mycelium ensure a firm and solid structure to penetrate the host. So this, if you look into the cell wall structure also, it'll be easy for them to penetrate. So basically, what we will we have to understand here is, if you are going to use any filamental fungi, then the penetration of your organism into the substrate, the surface of your substrate, is going to be very, very easy. And because of this penetration, the nutrients from the substrate can be fully utilized. So once an organism is completely getting nutrients, it will obviously, the growth of the organism will be very, very good. So with keeping all these aspects in mind, like using filamental fungi is going to be very good for your solid state fermentation. So here also I have just mentioned to you the hyphal growth for spore, from spore. So how this hyphae grows, I've just given this picture, have a look at it. So now we'll move on to the schematic diagram of interaction between the filamental fungi and the solid state substrate surface. So the surface of your substrate and your fungi, how the interaction is going to be there. So you can just see the hyphal is just above the surface of your substrate. Okay, so the penetration that is happening there in the, you can see this S region, substrate pore. So you can see the interaction, how through this hyphae the fungi can utilize all the nutrients. So this fungi is attaching to the surface of the substrate surface so easily, and the interaction is maximum. So because of this, the nutrients that are available within the substrate is fully utilized by the organism. So once they get good nutrients, they are going to grow healthy. Fine. So I've just given a schematic representation here for you to understand how the interaction between the filamental fungi and the substrate happens. Okay. So now, example of microorganisms that are involved in solid state fermentation process. So I've just given few examples. That is your for fungi, I've given Aspergillus, I've given Aspergillus niger, I've given Trichoderma, all these organisms, and for what process they are used. I've just mentioned few process with the example for this example. And in case of bacteria, you can see this Clostridium species, Streptococcus, Bacillus species there. And in case of yeast, you can find the Saccharomyces cerevisiae. So this you can see there for food and ethanol, for ethanol, bioethanol process here. And okay, we spoke about microorganisms here. So we have concluded that fungi is going to be the best option for your solid state fermentation process. So what are the differences between this fungi and yeast? So one major difference that you can tell is that yeasts are unicellular and your fungi are multicellular. You can majorly find some differences in their structure. So this is a major difference between your yeast and your fungi, and that's why fungi is said to be good for this solid state fermentation process. Okay. Right. So next is examples on uses of this filamental fungi in solid state fermentation process. So what is koji? So it is nothing like this. It is a cooked rice. Okay, this cooked rice is that has been inoculated with Aspergillus oryzae, a mold that's widespread in Japan, also named kojikin or koji mold. The rice is then placed in wooden trays, left to ferment in a warm and humid environment for up to 50 hours, and the result is essentially moldy rice. So this is nothing like, can you see the tray that they have arranged there? So as I said, like if you're going to do it in a lab scale, you can do it in a clinical flask. If you are planning to do it in a pilot scale, you can use this larger tray reactors. So these are called tray reactors. This is like nothing, the cooked rice. Okay, they are going to add Aspergillus oryzae fungi with into this cooked rice, and they are going to let it ferment for 50 hours. So what happens? You can just give a moldy rice there. Because this fungi is said to be a moldy slime, isn't it? So it's going to obviously, the growth of this organism will give a moldy slime. The fungi digest starch and proteins and break them down into sugars and amino acids. Because of this, it can be used as a starter for a number of fermented Japanese food products. So this fermented product can be used in soya sauce and all. It's going to be a starter Okay, it is again going to be kind of an inoculum for other fermented foods to process. Okay, so this is one example, and the other example is this tempe. So this is an Indonesian fermented food. I've just given the pictures of tempe. Growth of rhizopus on cooked soybeans. Okay, so on cooked soybeans, they are just going to inoculate with this rhizopus, and this bean is being dehulled and soaked overnight, and it is cooked and allowed to cool, and then this culture is added to it. So you can just see it in the, there'll be you can see the package here, packed tempeh product there. So this is also one good example for your solid state fermentation. So we are just looking into few examples that industrially available products which utilizes this solid state fermentation process. So we are just looking into that. So next is your tape or tape is a cassava, white rice or glutinous rice. So this ragi is solid culture medium grown with different types of filamentous fungi. So you can just see the picture of this product here. So for this ragi, you can see this. It is in Malaysia and Indonesia. So you can just see the images of it. So these are all few examples that you can see for the solid state fermentation process. Okay. Next is your, so we have completely seen the selection of microorganisms, like what are all the microorganisms that are available and on what basis, on what criteria that we are going to select the microorganisms. Okay, so it is based on the application. You can choose different microorganisms, and mostly used microorganism is said to be fungi. So why fungi is said to be most widely used? It is because of its structure. So it's hyphae. Through that, it is going to easily penetrate into the surface of your substrate, and it will be easy for the organism to utilize all the nutrients that are present in the substrate. Okay, so this is the major selection criteria for microorganisms. Okay, what is next now? Substrate characteristics and pre-treatment. Okay, so how you going to characterize your substrate, on what basis this, what are the basic characters of your substrate, and how you are going to do this pre-treatment process. Okay, so now the first we will look into the characteristic part of your substrate. Fine. So generally, substrate for solid state fermentation are products from agriculture or byproducts of agro industries which are cheap and abundant. So example is your cellulose, starch, pectin, lignocellulose, and fibers. So the major characteristic of your substrate, that the substrate which you are going to use for your solid state fermentation is that it will be maximum waste that is generated from agriculture, or sometimes it may be a byproduct that is from agro industries. So since they are waste, they are they are no more involved in that process, they are considered to be waste, and they're not going to use this biomass anywhere for that process. So because of that, we are going to reuse it. So since it is a waste, it is obviously considered to be very cheap, and they'll be abundant in nature. So for that example is your cellulose. So what are all the different substrate, or you can also call it as biomass, that is involved for solid state fermentation? Is that I've just given few pictures here. Can you see the waste from sugar industries, so sugarcane bagasse and agricultural waste and all? After harvesting, there will be definitely some waste that you're just going to throw it away. So why can't we use those waste for our process, right? So I've just given few pictures of the biomass. Okay, now coming back to the characteristics of your substrate. The solid state substrate is a porous matrix which can be biodegradable with large surface areas of microbial growth. Yes, so if your substrate is going to be highly porous, it can easily take up the media that you're going to pour into it. Okay, so if it is very highly porous, it can easily take up the media that you're going to pour into it. Once your substrate can easily take up all the media that you're going to pour into it, what is the use of that? So you will get the surface, this a large surface area, one that is going to it's going to be porous. It will definitely have a large surface area, and it will be easy for your microorganisms to widely spread and grow. So that will be one major characteristic for your biomass. Okay. So then the solid matrix should not be contaminated by inhibitors or microbial activities and should contain available microbial foodstuffs such as carbohydrates, nitrogen sources, and mineral salts. Yes, obviously. So as I said, your microorganisms are going to utilize the nutrients that are present in your biomass, and then they're going to grow. So obviously your biomass should have this major characteristic that all the necessary nutrients should be there in that biomass for the supporting the microbial growth. So here I've mentioned carbohydrates, that is your cellulose, starch, and other sugars in them, and your nitrogen sources, ammonia, urea, and peptides, and other minerals that are required for us. So see, like you're going to pour some media into it, isn't it? So this media will definitely have a carbon source and nitrogen source, and other mineral salts will be there in it, right? So likewise, this biomass should also have all the required nutrients into it. Only then your microorganism will utilize all these nutrients and it will grow, right? So for to give a just about this class, we have covered what is basic introduction. What is a solid state fermentation? So why do we call a solid state fermentation? It is because your solid state fermentation is going to happen within the solid substrate. Okay. So in the introduction class about enzymes, we saw what are all the different types of fermentation that is available. One is your solid state fermentation, and the other one is your submerged fermentation. So about submerged fermentation, we have already discussed a lot about the submerged fermentation. So the solid state fermentation, it is, as I said, it's going to happen on any solid surface. And what is the solid state fermentation? It has two phases. One is your microorganism is going to grow there, and the second is a product is going to form. Product formation. It is focused on growth of your microorganism as well as a product formation. Correct. So how this solid state fermentation is happening? So there are certain criteria. So based on these criteria, we can choose the the process. You can easily design a process. First, we'll know what are all the parameters that that are involved in it, and what are all the selection criteria and all. We need to know about that. Only then you can easily design a process and you can proceed with that. So the solid state fermentation can we saw how to do it in a lab scale, how to do it in how it is done in a pilot scale, and how it is done on a large scale. And then we came into the major factors that are influencing the solid state fermentation. So under that, we saw the selection criteria of microorganisms. So under selection criteria of microorganisms, we saw what are all the different microorganisms that are available. What are all the different microorganisms? Your yeast, fungi, and bacteria. So if in case of bacteria, this food processing bacteria which are involved in food processing are majorly used. And in case of yeast, it is used for converting your sugar into ethanol. That we spoke in application of enzyme in biofuel industries, isn't it? So then we spoke about fungi. So when compared to all the other microorganisms, this fungi is said to be one of the majorly used microorganism for solid state fermentation. Correct. So why we are using this fungi for solid state fermentation? Because fungi, the structure of fungi is going to support more for this process. The penetration of your fungi in case of your substrate is going to be very good because of its structure, the the filament structure. It can easily penetrate to the surface of your substrate. So once it can easily penetrate to the surface of your substrate, it will definitely be easy for the microorganism to utilize the nutrients that are present in it. Correct. So all these criteria, fungi is considered to be one major microorganism that can be used for this solid state fermentation. Correct. Then now we started with the, what are all the characteristics of the substrate. So first we saw this substrate should be it is coming from agro industrial waste. So since it is agro industrial waste, it is said to be very cheaper, and since they are waste, they in a everyday process there are a lot of waste will be generated, so they are abundant. And what are all the components there should be there in your substrate? So it should be cellulose, starch, pectin, lignocellulose, fibers, and whatever be the resources. And next we saw the it should be very porous. That is, your substrate should be very porous. Why it should be porous? Because only if it is very porous, it can easily absorb the media, and the growth of the organism will be very even. Once the media is getting mixed with the substrate properly, so the nutrients will be evenly distributed. Once the nutrient is evenly distributed, the growth of the organism will also be evenly fair. So you'll get a good amount of biomass if you start with the process. Okay? And what are all the different nutrients that should be available? So your substrate should have uh, carbohydrates, it should have nitrogenous sources and some minerals into it. Only then, it can easily grow. And your substrate should not, get easily contaminated. Okay. And apart from all this, we need to discuss about this also. That is how far your substrate is acting to the process, and it should not release much inhibitory products out from it. So the inhibitory products that is getting released from your process should be like very minimal, because if your substrate is going to release a lot of inhibitory products out, so that will definitely going to, they are called inhibitory products because they are going to inhibit the production of the major product. Okay, so they shouldn't go and cause any hindrance for the production of your product. Your product should form smoothly. So in keeping all these criteria in mind, you need to choose your biomass. And I have also given few pictures of your biomass, sugarcane bagasse and rice hulls, and so these are all different biomasses that can be used for your solid state fermentation process. So I think this lecture have covered the major topics. So in next class, we'll be talking about the other parameters, the major factors that are influencing the solid state fermentation. We're going to talk about what are the advantages of solid state fermentation, what are all the disadvantages of solid state fermentation, and all. So thank you so much for attending the lecture.